Chris Ashcraft. Chris has been here a number of times now speaking on different topics related to creation. You may know Chris is a teacher, science teacher at the Cedar Park school system. So he looks at science and the Bible every day. That's part of his teaching. And so he's very well qualified to address the topic at hand. He's also the founder of the northwestcreation.net website, which have information on any topic you might think about on creation. A lot of good detail that supports the model of creation. Chris is going to talk on this topic of what Darwin didn't know. Remember, Darwin wrote his book 150 years ago. He didn't know all the things that we know today. And so the question is, does what we know today support his theory, or does it disprove it? And the answer is going to be, and Chris, come on up and explain that. I'm going to pass the sign-up sheet around. I forgot to do that at the beginning, but if you're not on our mailing list yet, just add your name to that. Chris?
Thank you, Heinz. All right, so we're going to talk about evolution tonight. My goal is kind of twofold. One, I want to help you understand what evolution really is, how it takes place, how it is that animals adapt to the various environments out there, and then to point out a number of problems with it as well. But if you don't really know much about evolution, and if you don't know what a species is or what speciation is, then there will be some new terms there for you. And hopefully you'll come away with this with some new knowledge and better understanding about what evolution is, what can we accept and what we shouldn't accept, you know. So in starting there, on our definition of what evolution is, it's important to understand that it is a necessary theory that's been put forth by scientists who hold to an atheistic worldview. Okay? Scientists are doing their best to explain how our world came to exist through purely natural processes. So I'm not going to say that all scientists are atheists, because certainly they're not, but the majority of scientists today are atheist, and the science that's being taught in our public schools certainly is a science that's based on atheism. They are doing their best to explain, forgive me, always have my technology issues. I'm on a little remote working up here. I'm like, it's not changing slides, it's not changing slides. Oh, well, you got to plug something in. So, okay. So, evolution is an attempt by scientists to hold to an atheistic worldview, an attempt to explain how our world came to existence without God. That's what evolution is. It's an alternative to the creation account in the Bible. It's an alternative explanation of how we came to be here, how this world came to be here. It's a theory that's based on atheism, the view that there is no God. We hold to theism. They hold to atheism, meaning the view or belief that there is no God. From this worldview of atheism comes naturalism. Scientists today are dedicated to naturalism, to explaining how all this came about through purely natural processes. They would argue that every physical phenomenon must be explained as occurring through a natural process, and the origin of everything we see, the origin of the world, the origin of life on earth, all must be explained as coming into existence through natural processes alone. And again, this is in stark contrast with the Bible. The Bible's history of how this world came to be is one of an acts of supernatural creation by God. The naturalism itself, this philosophical view of naturalism, is itself in conflict with the Bible. From naturalism, when applied to the living world, this is where we get evolution. So evolution is an attempt to explain how the living world came to be through purely natural processes. That's what it is. It's important to understand what it is, so that we can better analyze it for its various components. And of course, when evolution is applied to the human condition, to humanity, that's where we get humanism. The view that humans are just another animal, that there is no God. We come about through purely natural processes, so we can decide for ourselves what is right and what is wrong. And I mean, there are various reasons for challenging evolution or teaching against these theories, but I think probably the most significant aspect of this can be defined by an evolutionist. William Provine is professor at Cornell University, and he's in the Department of Evolutionary Biology, and he describes the outworkings or the consequences of Darwinism or the evolutionary worldview. Listen to William Provine.
Let me summarize my views on what modern evolutionary biology tells us loud and clear, and I must say that these are basically Darwin's views. There are no gods, no purposive forces of any kind, no life after death. When I die, I am absolutely certain that I'm going to be completely dead. That's just all, that's going to be the end of me. There is no ultimate foundation for ethics, no ultimate meaning in life, and no free will for humans either.
Well, the ultimate consequence of the evolutionary worldview is this. Not only do we decide for ourselves what is right and what is wrong, but there's no meaning in life. No free will. There's no free will. They argue that we're just a bag of chemicals and we're, you know, what we're doing is just instinctive to us. We're acting out our instincts, our animal instincts. How could you say that someone is doing something right or what is wrong when it's all instinctive? In reality, they say that there's no free will. We're just following through with our animal instincts. That's all it is. So let's talk about evolution, what I would call Darwinism, just a synonym for evolution today. Darwin published his seminal book, On the Origin of Species, in 1859, which included two main principles. One called the principle of descent with modification, as well as a mechanism to explain how organisms change through time, what he called natural selection. So the general theory of evolution holds that all organisms on Earth have descended from one common ancestral cell. They call that original cell the last universal ancestor. But all life on Earth, they believe, has descended through modification from one ancestral cell that evolved a long time ago. The origin of life at this point has been postulated as occurring about 3.9 billion years ago here on Earth. In 1937, he sketched a diagram of an evolutionary tree in his first notebook titled The Transmutation of Species. Today, this notebook shown here is on display at the Museum of Natural History in Manhattan. Interpretations of his handwriting, I'll display here for you so you can read a little bit of what he thought. Up in the upper left-hand corner of his notebook, you can see where he sketched, I think. I think, he said, case must be that one generation then should have as many living as now. To do this and to have as many species in same genus requires extinction. Thus between A and B there's an immense gap of relation. Between C and B there's the finest gradation. Between B and D there's rather greater gradation. Thus genera would be formed bearing relation to ancient types with several distinct forms. What he's ultimately talking about here, what he's theorizing about is the big puzzle for evolution, and that how it is that we have several distinct groups, numerous distinct groups of organisms that are separated from one another by tremendous gaps. He was trying to speak to this within this. How do we explain these gaps? Because Darwin's theory ultimately was numerous successive slight modifications. You should see a slow and gradual increase in the number of organisms on Earth, the amount of diversity seen on the organisms on Earth, but we see distinct groups of organisms, which is completely consistent with the biblical view, where God created several kinds of plants and several kinds of animals. Genesis 1, let the earth bring forth living creatures after their kind, cattle and creeping things and beasts of the earth after their kind. And it was so. God made the beasts of the earth after their kind, and the cattle after their kind, and everything that creeps along the ground after its kind. And God saw that it was good. And these kinds are also the kinds that were brought on board Noah's ark, mentioned there as well. But I will establish my covenant with you, God said to Noah, and you will enter the ark, you and your sons and your wife and your sons' wives with you, and every living thing of all flesh. You shall bring two of every kind into the ark to keep them alive with you. They should be male and female, of the birds after their kind, and the animals after their kind, and of the creeping thing of the ground after its kind. Two of every kind will come to you to be kept alive. The Bible speaks of many different kinds of animals. This is completely consistent with what we see in living populations or in the fossil record. We see distinct groups of organisms, different kinds. Prentice Hall Biology is arguably the most widely used biology book used in U.S. public schools today. This is how they describe Evolution. They say Darwin argued that living things have been evolving on Earth for millions of years. Evidence for this process could be found in the fossil record, the geographical distribution of living species, homologous structures of living organisms, and similarities in early development or embryology. What I'd like to do is try to tackle some of these. Now, there are many proofs for evolution. There are many evidentiary supports that are brought forth. I won't have time to go into the fossil record today, so we will stick just with the evidence that comes from living population, but we will want to look at some of these, the diversity and homology and embryology in particular. We'll want to look at these and see if there really is support for evolution there, or are there problems there. Okay, let's look first at Darwin's theories of descent with modification and natural selection. This ultimate theory was derived from his own breeding experiments with pigeons. Darwin grew up in England at a time when pigeon breeding was a very, very popular hobby. All the way from poor to the very rich could be involved in pigeon breeding. Very expensive breeds, of course, could only be attained by the rich, but pigeons were found all over the place. You could pick a couple of them up, throw you together a little ramshackle pigeon coop, and next thing you know, you got your little population of pigeons and can start doing some breeding with them if you want. Darwin published a book, in fact, in 1968, describing the pigeon breeding history that had taken place there in England. But from numerous pigeons like the rock pigeon, or the one shown here is actually a rock pigeon. From the rock pigeon, numerous distinct breeds of pigeons have been developed over the years. And again, Darwin published a book in 1968 titled Variation of Animal and Plants Under Domestication, where he charted in this book the breeding history, or what we might call a genealogy of domestic pigeon breeds. In this book, he states that the domestic breeds differ as much as any genera of wild pigeons, in the quote there. But in fact, the domestic pigeon breeds differ much more than do natural breeds. And this is the case with all domestic populations. I used to have a Persian cat. I had a buddy of mine give me a full-blood Persian cat once, and that cat was a high-maintenance cat, I'll tell you what. Not only did you have to clean his eyes out all the time, but his coat was so dense that it would mat up. And it would mat to the point where it would limit its mobility eventually if I wasn't constantly brushing it. And if I didn't maintain the brushing well enough, that eventually I'd have to come through with the shears and literally just cut whole sheets of hair off of them at one point in time. I mean, I do not believe a cat like my Persian could survive in the wild. There's a much greater diversity there that in these domestic populations than we see in the wild, because many of them simply couldn't survive without our constant grooming and these kinds of things. These domestic breeding activities provide us with a good measure of the amount of variability that is available to these type of organisms. And Darwin postulated that the same type of selection was occurring in nature. We create breeds by selectively breeding them. Darwin argued that nature was doing its own selective breeding, what he called natural selection. Rather than artificial selection like we do in these breeding programs, nature could select as well. But it selects due to the differing abilities to survive within variations we see in a population. And if you look at, if you think about the various breeds that you're familiar with, or the various types of dogs, or various types of cats, you consider any of these kind of populations, you can imagine variations that would be more or less successful in different areas. The size of the animal alone is very significant if you're considering predator-prey interaction. How big you are or how fast you are could affect your ability to survive in a predator-prey kind of interaction situation, or even the type of hair. The coat that an animal is wearing could greatly change how successful they are in various habitats. You want a really thin coat if you're in a really hot habitat, right? But if you're up in the Arctic region, you need a coat that's going to help you survive the Arctic. Okay? Nature can select from these. So consider in a population, you have offsprings of pups that are various sizes, various speeds, or can run at various speeds, have various different kinds of coats. Within this population, some of those pups or kittens or whatever you want to talk about are going to be have a greater advantage than others will be to survive in a natural environment. And this was his main mechanism, what he called natural selection. Darwin also later drew from observations he made while on his famous voyage out to the Galapagos Islands. Darwin hired himself on to be a naturalist, which is a synonym, was a synonym then, and still to some degree a synonym today, for a scientist. Darwin hired himself on the board of the HMS Beagle to be an unpaid naturalist, which is what we call scientists today, because science is dedicated to naturalism. So we call them just naturalists. Or that's what we call our science museums natural history museums, right? Or why we call the world that's out there nature, because that's all natural. But I argue that's not natural. That's not, I don't even like to call it nature anymore, because that's the creation. Okay? But on Darwin's famous voyage at the Galapagos Islands, what he observed out there were a number of pigeon species. Finch species. There were a bunch of these finches, the little birds that were on the various islands of the Galapagos. And when he got back and looked over his notes, he recognized something was significant there, that on the various islands, the beak of the finch on the various islands were different. And it was argued that, well, on this one island, they were eating something a little bit different than they were on this island, which is probably very correct. On this island, if the only food source is a seed, you need a beak that's nice and formidable to crack through those shells of that seed. Or if your beak is, if the only food on this island are little worms, and you have to drill into a plant to get those little worms out or something, a beak that's nice and thin to get in there will help you get that food. But this was, and so Darwin used this example in his book on the origin of species, these finches. But is this really evolution? The fact that the beaks of the finches changed through time to help them adapt to the food source on the various islands, yeah, I think we can accept that. No problem there. I think we can accept natural selection. No problem there. But is this really evolution? Is it evolution, the big picture of evolution? Do animals evolve through time? Yes. But the big picture of evolution, again, what we call Darwinism, is that all organisms on Earth have descended from one common ancestor. At the end of the story on the finches, they're still just finches. They have slightly different beaks, but this is a long way away from providing the kind of evidence that this big story of evolution really needs. Let's listen to a few comments from Jonathan Wells. This is from the documentary Icons of Evolution.
Another icon of evolution are some species of birds on the Galapagos Islands that are very similar except for the size and shape of their beaks. And it's thought, and it's quite possible, that they evolved from a common ancestor because of having to eat different foods on the different islands.
In 1994, journalist Jonathan Weiner published The Beak of the Finch, a book about research into the Galapagos finches. It won the Pulitzer Prize. Weiner argued that the finches represent the best and most detailed demonstration to date of the power of Darwin's process. Similar claims are made in many biology textbooks.
The Galapagos finches are a spectacular example of evolution. When the food quality, in other words, the kind of food that was available, changed as a result of a drought or a particularly rainy year or something like that, over the course of just a couple of years, they could see dramatic changes in the beak sizes in various populations. Natural selection can drive changes of a structure, like the beak, in one direction in response to selective pressure, not just fast enough to account for what would be required for Darwin's theories, but actually 50 to 100 times faster than that.
A severe drought could cause many of the finches to die and leave only those with larger beaks. So in the following generation, the average beak size was increased. And some textbooks extrapolate this over 200 years or so and say that a few of these events strung together could transform these finches into a new species by making their beaks larger.
Look, the stuff about finch beaks is certainly interesting. Let's not confuse ourselves about that. The question is, can it be extrapolated, or does it represent cyclic variation?
What the books fail to mention is that as soon as the rains came back, the average beak size returned to normal. There was no net evolution. What we're really seeing is just one species oscillating back and forth with no real evolutionary change. So The evidence is exaggerated to make it appear to support Darwin's theory in a way that it really doesn't.
The contrary may be true. We may be seeing the development of entirely new species. The Galapagos finch starts off as a finch, and within 100 million years there'll be a Galapagos elephant. Could be. But we need a whole lot more by way of evidence than a couple of nutty journalists going down there looking at finch beaks and writing a Pulitzer Prize winning book. A whole lot more if this is to be serious science. I mean, this doesn't even pass the threshold of anecdote. Finch beaks change in size. Yeah, they do. They change in shape too. It seems to be correlated with seasons. There seems to be a regress back toward the mean when the seasons change again. If this is the part of a spectacular evolutionary extrapolation, let's have additional reasons for thinking that.
The changes are temporary, they oscillate back and forth, and they don't go anywhere. So, as evidence for the origin of species, Darwin's finches really don't work.
Still just finches. So although Darwin's book that he published back in 1859 was called On the Origin of Species, he never actually talked about the origin of species. What he actually talked about was how species change through time and provided the mechanism called natural selection. But what is a species? So Ernst Mayr, the eminent evolutionary biologist Ernst Mayr, eventually established the definition of species that most people hold to today, and that a species is a naturally occurring breeding group that's reproductively isolated from other such groups. So it's a population that's breeding, and they are separated from animals of the same kind from breeding. So it's a separate breeding population, a breeding population that's separate from another population elsewhere. We call these species, and a speciation event would be the time when a population becomes separate or splits into two populations. You could define that as the moment when a new species develops. Okay, the most common type of evolution is due to a simple geographic separation. It's what they call allopatric speciation. But through time, what can happen, a population can start to spread out and fill an area, and in the process of migrating and filling an area like this, a population can become separate, can become splintered. Okay, and when when two populations are separated from one another like this, they will start to adapt to different regions. And the adaptations that develop over time can lead eventually to genetic differences that prevent the two populations from mating once they reunite. So like in this diagram, these two populations went around different sides of the lake. By the time they eventually were reunited back on the other side of the lake, they could no longer breed together. Enough genetic differences can develop that they're no longer able to produce fertile offspring. It's a true speciation event. And we know that this has happened in several populations. For example, the California Estatina, a type of salamander there in California, has speciated through a similar mechanism. Okay? But interestingly, we know that this process actually begins with choice. Although it is true that the two populations can become genetically incompatible, what frequently happens when the two populations are reunited, they instinctively remain separate. They will only breed with their own group. Even if the two populations start to inhabit the same area, they remain instinctively separated. Instinct, it occurs through it's an innate behavior, a behavior that's designed to preserve specializations. Because when organisms adapt to an environment, those adaptations involve genetic changes, and you don't want to lose those. If two populations reunited and bred back together, you could lose specializations. Consider the specializations, for example, the polar bear. If the polar bear bred with the brown bear, all those polar bear specializations will be lost to that first generation. Okay, so it's important for them to be preserved in this way, and they apparently are through instinct. And we typically use trees like this. You'll see these kind of trees to diagram the historical relationship between organisms in a population. These trees show the relative times of speciation. The branch points would be the point when two species had developed, and the relative morphological or structural changes that took place between the intervening period are also shown in relative distances. So we tend to see these a lot, what we call a phylogenetic tree. Okay? This one just showing the evolution of a few species, but we can see you can find these that have every organism on Earth fit into that same phylogenetic tree. Now, from the biblical narrative of the flood, we know that there was only one breeding pair of unclean animals on board the ark. The dog and the cats both are unclean animals. They're predators. The clean animals were the obligate herbivores, like your cows and your deers and things. 7 pairs of every clean animal was aboard the ark, but there was only 1 pair of unclean animals, like the canines. So we know that all of the living canines have evolved or descended from 1 pair of canines that was on board the ark. 1 pair. So the wolves and your jackals and your coyotes, all of these, their foxes have all evolved from 1 pair of canines that was on board the ark. Okay? Or all of the various domestic breeds that we see of canines. All of these have been isolated from the gray wolf. Historically, this is pretty well verified, that at some point we humans domesticated the gray wolf, and that living in domestication, this gray wolf started to express some variations that breeders then used to create all the various dog breeds that we have today. Okay? But this artificial selection, this breeding history that we have, has been used, and or the small scale evolution that we see happening in living populations is being used to support this grand scale evolution. Populations do change through time. The finch, the various finches that we see on the Galapagos Islands, we can accept that level of change. Or the development of the various dog breeds, we can accept this level of change. What might be termed microevolution, or what used to be termed microevolution, is this small-scale adaptational change that involves a change in gene frequency through time in those populations. But this small-scale change is what's being used or extrapolated into large-scale evolutionary process. It's being argued that the small-scale changes we see in these finch populations through time would enable this process to produce all the organisms we see on Earth today. This is what the theory of evolution really is. It's an attempt to explain small-scale change, or to extrapolate small-scale change into all the organisms on Earth. So the one phylogenetic tree that I showed you with a couple of speciation events illustrated, evolutionists have put together these large phylogenetic trees that show how all organisms on Earth are related according to their theory. But this is what we have a problem with. I don't think we have a problem with various finch species developing on the various islands, or that level of change is possible for organisms in a very short period of time. It's this grand perspective that we have a problem with. As well, we have a problem with the source for that variability. We'll talk about genetics here in a little bit and mutation, but that is also a place where we would take issue. Ernst Haeckel was an evangelical promoter of Darwinism in Europe, in Germany and specifically. Darwin believed that Haeckel's enthusiastic promotion of his theory was the primary reason why it was accepted in Europe. He became Darwin's chief European apostle, proclaiming the gospel of evolution with evangelical fervor, not only to the university intelligentsia, but to the common man by popular books, and to the working classes by lectures and rental halls. He was, in fact, the first person to draw an evolutionary tree, a phylogenetic tree, that included man. Up until this point in time, evolutionists had steered away from adding humanity to an evolutionary tree. Ernst Haeckel was the first person who added humans to the top of his evolutionary tree. He believed that the only major difference between apes and man was that men could speak and apes could not. He therefore postulated a missing link, which he called Pithecanthropus allalis, which basically means speechless ape man. And even had an artist, a guy by the name of Gabriel Max, draw up the imagined creature that you see here. Although there was not a scrap of evidence to support the details of this drawing. No evidence for this creature was ever found. It was a theoretical necessity. A contemporary of Ernst Haeckel, actually, Rudolf Virchow, famous as the founder of cellular pathology, and for many years was the president of the Berlin Anthropological Society, was scathing in his criticism of Ernst Haeckel. For Haeckel to have given a zoological name, he gave it a species name. To give a zoological name to a creature that had not one shred of supporting evidence was a great mockery of science to Virchow. Haeckel would eventually became notorious as the scientist who perpetuated fraud after fraud to promote the theory of evolution. Now, there are a number of evolutionists today that are critical of Darwinism or certain aspects of evolution, although much of that remains behind closed door. You met David Berlinski previously. He was one of the people talking in that clip on the finches. David Berlinski has a PhD from Princeton and has taught at Stanford, Rutgers, the City University of New York, and the University de Paris. Listen to some more comments from David Berlinski.
The claim that all skeptics about Darwinian orthodoxy or Christian fundamentalists stands refuted by me. It's obviously not true. I'm neither Christian nor a fundamentalist. But lots and lots of people are skeptical in the scientific community. I know dozens of mathematicians who scratch their head and say, you guys think this is the way life originated? It's absolutely a preposterous theory. And many, many very significant figures. John von Neumann, one of the great mathematicians of the 20th century, just laughed at Darwinian theory. He hooted at it. So it's perfectly absurd. This is a point in a polemical dispute. It's not a reasonable standard of criticism. Opposition to Darwinian theory is, I wouldn't say widespread, but there's a consistent group of people among mathematicians, among physicists, among some very good speculative biologists who simply don't accept it, don't even regard it as a scientific theory in any reasonable sense.
So there are a lot of people that are skeptical, but most of that skepticism is behind closed doors. If you're in a position of academia today or in science industry, you don't challenge Darwinism. Not in open forums, that's for sure. So let's talk about some things Darwin didn't know. Some of the things that weren't known when Darwin first published his theory. Darwin didn't know about heredity, what we might call genetics. The principles of genetics were established by Gregor Mendel, shown here, who was an Augustinian monk who is today considered the father of modern genetics. He did experiments while living in an Austrian monastery. In 1866, his work, titled Experiments on Plant Hybridization, was published in the Proceedings of Natural Research Society of Brunn. So this is close to a decade after Darwin published The Origin of Species. Mendel's work on establishing the principles of genetics was finally published. But the significance of his work wasn't recognized until close to the turn of the 20th century. But again, the father of genetics here, a Bible-believing creationist, right? A monk living in an Austrian monastery. But that is not an isolated incident. The fact that we have Bible-believing scientists who have made very significant contributions to science. It's arguable that the scientific revolution itself occurred because you had Bible-believing men that were looking into science from a creation point of view that made great strides during that period. The vast majority of the founding fathers of the various science disciplines believed in God. The vast majority of the founding fathers of our science disciplines were what today would be called creationists, and today they would be told they couldn't even be scientists. Because you can't be a scientist today without holding to naturalism. You believe in creation? No. You couldn't get published if that was known. What Gregor Mendel did, he did experiments with pea plants. So stick with me for a little bit because there's something significant I want to point out here. What he did, he did experiments with pea plants. He bought purebred, like a purebred pea plant that had yellow flowers, and bred that with a purebred pea plant that had white flowers. So he crossed these two plants together. Yellow flower and white flower, he crossed them together, and then he would look at the offspring. And these kind of breeding experiments, again, he was a monk, but these monks were also academics, and he was the monastery gardener as well as kept good statistical notes of his breeding histories, and he established many important foundation principles of genetics, like the principle of dominance. If you have two different traits, one of those tends to dominate the other trait. You breed a plant with a yellow flower together with a plant with a white flower, all the first generation may just be yellow flowers. The white flower disappears in that first generation because it was dominated by the other trait, okay? So he did these kind of experiments, and what he found was that in the offspring, there was always a very specific ratio of traits that were found in the offspring. A very specific ratio was always found. If he mated a dominant trait with a recessive trait, in the next generation, 3 of them had the dominant trait, 1 of them had the recessive trait. There was always a 3-to-1 ratio. It was always a very specific ratio of traits that were found in the offspring. And he even expanded this into including 2 different traits, like size of the plant and color of the flower. 2 different traits, and bred those plants together that had 2 different opposing traits. And he still got very specific numbers. 9, 3 to 3 to 1 is the ratio they got. 9 plants would have both dominant traits. 3 of them would have 1 dominant trait, 1 recessive trait. The other 3 would have 1 dominant trait, and the 2 would have both recessive traits. He always got very specific numbers. Very, very specific numbers, which illustrated well that the amount of variation available to these populations is fixed. It's very, very limited. It's limited to the number of genes that is possessed by that organism. The kind of variation we can see in populations is very limited, okay? And I almost want to go into something else, but I think it would be a little too challenging. At this point, one of the big unknowns, though, is where did that variation come from? Okay? In attempting to explain where all of the variation in the dog breeds came from, the evolutionists say that all of this variation came about through a history of gene mutations. Creationists say, no, the gray wolf already had all that variation, and breeders have just managed to isolate it. But there's one important thing that a lot of people failed to recognize, and that the gray wolf was a pure breed. Well, gray wolf only gave birth to wolf pups. After domestication, all of that variety seemed to have developed. There's some significant there. I'm going to leave that to you, though. Okay? Darwin also didn't know about DNA. They didn't know anything about DNA during Darwin's time. They knew that cells had several big macromolecules, proteins versus DNA. At this point in time, they were still debating on whether protein or DNA was the molecule that carried the inherited information that caused you to look the way you look. Okay? They didn't know anything about DNA. So let's talk a little about DNA and what it does. DNA is found in the nucleus of cells. The nucleus, that center compartment of the cell, is where we find our DNA. It's typically modeled like that little helical. It's what we call a double helix model of what DNA looks like. Or when you see them illustrated as a chromosome, you may see a long strand of DNA that looks like that. DNA, it exists in long strands we call chromosomes. Humans have 46 chromosomes. They range in size from 1.7 to 8.5 centimeters in length. In total, if you take every single chromosome in one of your cells and add them end to end, you'll have about 6 feet worth of DNA in every single cell in your body. It's a lot of DNA, a lot of information. And that chromosome is made up of individual reading sections called genes. Gene. And what the gene is an instruction that tells cells how to make proteins. That's what they are. DNA is information. It's an instruction that tells cells how to make proteins. Even Bill Gates says that by far the most sophisticated program around is DNA. It's a program. Everyone knows that it's coded information. And therein provides one of the most powerful arguments for creation that there is. Because in all human experience, information, in our experience, only comes from one source, and that's an intelligent mind. In all human experience, information comes only from an intelligent mind. When we found that information existed within cells, it should have led to the conclusion that cells had come from an intelligent mind. Because that's how science works. You use your knowledge and experience to inform your theories and reach conclusions. Based on all human experience, we should have reached the conclusion that DNA was also the result of an intelligent mind. So let me show you how it works. The DNA exists as a code that is read by the cell. It actually works its way down the DNA molecule, reading these 3 letters at a time. Okay? And this is a code that tells it what amino acid to use when making proteins. So if it finds UGC, it uses cysteine in making the protein. If CUG is found next, it'll insert leucine next. If AGU is next in the code, it'll insert serine next. If CGA is next, it'll insert. So these are the codes that tell it what amino acids to use when making a protein. Important proteins like, that's hemoglobin right there. The protein that's in your red blood cells that binds oxygen and delivers those to your tissues. The body's made up of proteins. That's what you are, is you're a big collection of proteins. Proteins are the machineries, all the machines that are working within your body. Those are all proteins. Proteins are also often the building blocks. You have a protein, a machinery that's making your hair, but your hair is made of pure protein. So they're the machinery, and in many cases, they're also the building blocks. Proteins are everything, they do everything. That's why life is possible in general, okay? But it's a tremendous amount of information, okay, that we see in this double helix. This is what a sequencing gel looks like. I've done many sequencing gels back when I was a grad student at Texas Tech, and this shows you what DNA sequencing information looks like. It's a bunch of letters, because the molecules that make up the DNA chain are begin with the letters A, T, C, and G. Okay, so we just abbreviate those molecules with letters A, T, C, and G. If every one of those molecules, if every letter on there is equivalent to the letters in our alphabet, then the amount of information in one human cell, the amount of information in one human cell is about equal to that information in 1,000 books. If every letter shown in that DNA sequencing information is the same as a letter in our book, then one human cell contains about as much information as there are in 1,000 books. Or a small pinhead-sized pile of DNA contains about as much information as there are in 500 stacks of books reaching the moon, or a single stack of books reaching the sun. It's an incredible amount of information, incredible information storage system. Okay? So where do evolutionists believe all this genetic information came from? Well, that's mutation. They say that randomly that genetic code can get changed. UV bombardment, people might know that if you expose yourself to too much ultraviolet light, you can get mutations, because those ultraviolet rays can strike your DNA and change one of those A's maybe into a C. It can mutate the DNA. And they argue that over time, the information that we now, all of this genetic information came about by mutation, by randomly changing the genetic code. This is what we call neo-Darwinism. Neo-Darwinism basically means new Darwinism. It is Darwin's theories now informed by information about DNA and genetics. So now we need to talk about variability informed by DNA. And that's where we come about by mutations, which in my opinion is one of the most ludicrous aspects of the whole theory of evolution. The claim that you can just randomly change information and get more information. I mean, it's an affront to reason. It's an affront to common sense to propose such a thing. And most geneticists that I'm familiar with, that I've talked to behind closed doors, will admit this. This is really just so ludicrous. Because what we know about mutations is that they're destructive. You randomly change information and it will destroy that information. That's just what happens. Many of our diseases today are in fact due to mutations. When you randomly change that genetic code, it will destroy the proteins that were supposed to be made by that code. If those proteins are important for life, you're dead. A lot of diseases are due to genetic changes, these kind of mutations. However, just to be a bit honest, when I was working at Texas Tech, I actually had an opportunity to look over plants that were being generated in a mutagenesis study. Scientists can intentionally mutagenize. They can actually expose cells or tissues to chemicals that we know will cause mutations to see what these mutations will produce. And I had a chance to look over a table full of ice plant mutants and all of the variation that was seen there. And it was an extraordinary amount of information. And we also know, if you're a good, if you're a good astute observer, that mutations every once in a while can produce some heroic consequences. That you can end up, get all these amazing skills and abilities, even grow claws out and you can fight off enemies. Mutations can be beneficial. They can be beneficial, or wait, this is just fiction. Oh, science fiction. That's what we call it, science fiction, because mutations don't produce beneficial changes. They produce death and disease. That's what they do. But the basic assertion is that you can randomly change information and get more information, not just more information, then better information. You know, randomly change the engineer's code that they use to make the big TVs, and maybe you throw rocks at your TV and maybe you get a flat screen eventually. I know I still got 2 of those big TVs at my house. One of them so big I can hardly carry it. And I'm strong too, but I can just barely wield that thing. I'm just hoping it'll break, you know. I use what I got. You know, use what you have, you know, and don't buy new stuff just because new stuff comes out, you know. But man, I'm kind of hoping that TV breaks. well, someone come to my house and kick that TV over or something, because I have to keep using it as long as it's there, you know. And it's a moral dilemma. Okay. Next, Darwin also didn't know about molecular machines. In the Origin of Species, Darwin said this. He said, if it could be demonstrated that any complex organ existed that could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down, but I can find out no such case. And this is a very, very telling statement. But because Charles, I tell you now, we have now numerous examples of molecular machines that simply could not develop through numerous successive slight modifications. We call these irreducibly complex. These molecular machines are so complex, you can't reduce them into individual components that serve a function. For molecular machines to work like this is actually a kinesin that's walking a package that it has down a microtubule. It's a little walking robot that used to transport materials from one place to another. Molecular machines like this don't function unless every single piece is there and every single piece is assembled. You cannot get a machine, you cannot develop a machine through numerous successive slight modifications and have it functional at every stage along the process. That's what would happen, that would have to happen for evolution to be true. That molecular machine needs to be functional at every single step of its development, so that at every single step of its development, it's more beneficial. Now, one more thing has been added, it's still more beneficial, even more beneficial. One more thing is added to it, now it's even more beneficial. But these molecular machines don't work. You remove one component, doesn't work, just lies stiff. Or look at this one. This is some bacteria swimming around. This is the bacteria that are swimming around with a little tail they have called a flagellum. Now, I did some calculations on this. These are bacteria. If you didn't know, little bacteria, and I'll show you what they're using to swim with. It's called a bacterial flagellum. But they're moving the little motor that they have enables them to swim at close to 60 cell lengths per second, which, when you break that down to larger size animals, means that they're traveling at about 164 miles an hour. And what are they using to travel at 164 miles per hour? They have an outboard motor. What's called a bacterial flagellum actually is embedded within the membrane of the bacteria, just showing all the components and the order at which all of these are assembled. This is the actual assembly order of the flagellar motor that's there in the cell membrane, and then a long filament, kind of the outboard motor part of this, is assembled at the very end. And I should point out that the bacterial flagellum here is made up of 25 different proteins. Each of them is in multiple copies ranging from just a few, or for others, tens of thousands. Each of these is an individual protein. Each of those little lumps on there is a separate protein. And this one, flagellin, the one that's used to create the large filament, exists in tens of thousands of units. You'll see, you really get a better perspective of how long this is. Once this thing finally speeds up, it'll show you the full length of the flagellum, going into a little detail here on how this is assembled. But it'll speed up. Eventually. I'm getting impatient. Getting impatient. All right, there it goes. So that's a lot of proteins to make up something like this. In the scientific journal Cell, flagellar expert David Rosier says the flagellum resembles a machine designed by humans. It is, in fact, the world's smallest rotary propulsion system. It has all of the components we use in our own motors. It has a rotor, a stator, it has a U-joint. It then has its little fan blade there, that long filament that serves as a fan blade. It's a motor. There's no way around that. That's what it is a motor. And in all human experiences, motors come from only one source, and that's an intelligent mind. When we found motors there within the cell, it should have led to the conclusion that those motors were also probably the creation of an intelligent mind, because that's how reasonable people work. But Francis Crick was one of the co-discoverers of the DNA helix. He was one of the 2 people that won the Nobel Prize for discovering the shape of the DNA molecule. He admits to seeing evidence of design, but says that biologists must constantly keep in mind that what they see was not designed, but rather evolved. Ernest Haeckel, who we met previously, described cells that he named monarans as being very primitive protocells. He says, these monarans are not composed of any organs at all, but consist entirely of shapeless, simple, homogeneous matter. They're nothing more than a shapeless mobile lump of mucus or slime consisting of albuminous combinations of carbon. Well, but where did Ernest Haeckel see this cell that he described as a shapeless little lump of mucus or slime? I mean, it's really easy to envision the origin of the first cell if that first cell is nothing more than a shapeless little lump of mucus or slime, or if that's all cells are, it's easy to see how something like that could develop. Because Darwin envisioned the first cell to have evolved in a warm little pond, as he described it, and most theories have held to something similar. But where did he see this first little cell? They were seen only as his imagination. They're, again, a theoretical necessity. However, the fact that he had never seen them didn't stop him from making more than 30 drawings of these imaginary monarans, which, along with 73 pages of speculations, were published in the prestigious scientific journal in 1968. He also gave them scientific names, such as Protoamoeba primitiva, the primitive amoeba that came before, Protoamoeba primitiva. Because, a primitive protoamoeba must have existed before the more complex amoebas we find today, so why not just go ahead and make it up? Make it up, have someone do drawings for you. But I tell you, the extent of the detail in his drawings on these monarans shown here is the measure of his fraud. And if you don't know, monaran was the original name of the bacterial kingdom. That's what he named monarans, were these little protocells. That ended up becoming the name for the bacterial kingdom, until more recently they split it into archaebacteria and eubacteria. But when I was in college, we learned the name of the bacterial kingdom as Monera. That's what it was. Again, the height of his deception is fully illustrated by the fact that he made such detailed drawings, detailed and accurate drawings, because he knew well that cells like that did not exist, or he knew of the complex nature of cells. He actually had published plates made on diatoms. These are the shells, if you were, of single-cell organisms. These are diatoms, or if you know what diatomaceous earth is, it's earth that's rich in diatoms. Okay? But they're beautiful symmetric forms. Beautiful symmetric forms shown for these creatures. But and Ernest Haeckel knew all about these. He actually worked at Jena University in Germany that had the top technology of his day for looking at microbes, and he had detailed images cast of these. These are diagrams that Ernest Haeckel made of diatoms. A couple of plates that he made in his book he titled Art Forms in Nature. They recognize that there's art in nature, but not that there's an artisan behind it all. And then, interestingly, they can see these things but not see them but not see them, not see them. He had access again to the advanced technologies and had plates made up of diatoms. He knew that, and these are not shapeless little lumps. These are the simplest cells that we know of, these diatoms, single-cell organisms like this, it's antithesis, the exact opposite of shapeless. Exact opposite of shapeless. It's something else. I mean, God did put a lot of evidence out there. Remember Romans 1:20? For since the creation of the world, God's visible qualities, his eternal power and divine nature, clearly seen in what has been made. So that men are without excuse, and it's there. All you have to do is look. And remember the monarans that Haeckel imagined that were originally, that's the name of the bacterial name that was originally given to the bacterial kingdom. And remember, it's bacteria that possess what is the most complex molecular machine we know of. It is a bacteria. The simplest cell we know of has the most complex molecular machine we're familiar with, the bacteria. That's a bacterial flagellum. That's the organism Haeckel was really talking about as far as his little monarans. It was Charles Darwin who first proposed an explanation for life's origin that complemented his evolutionary theory of life. In a letter written in 1871 to a friend of his, a guy by the name of Joseph Hooker, Darwin envisioned it occurring in a warm little pond. It is often said that all the conditions for the first production of a living organism are present, which could have ever been present, but if, and oh what a big if, he says, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes. At the present day, such matter would be instantaneously devoured or absorbed, which would not have been the case before living creatures were formed. Little optimistic to propose, though, that a cell could form in some warm little pond, because today we cannot even establish that a protein can form in a warm little pond. That a single macromolecule used by cells can form in a warm little pond. Proteins, carbohydrates, lipid, nucleic acids, you have a bunch of big macromolecules that all had to form spontaneously before a cell would form. There's big steps have to occur here. What Darwin didn't know about were the incredible odds on getting the origin of life to actually get going. So one of the things we now know is that you have to have not only these big macromolecules, all have to be there, but you have to have DNA to make proteins. We have to have proteins to make DNA. They all have to be there, and they have to be there at the exact same time. You can't just start with a protein and then add one more protein and eventually get up to a cell. You know, to have a cell, you have to have all of these molecules all there at the exact same time. So let's just look at the basic probability formulation here on getting one small chain of amino acids to form, okay? Now, I can't really think of any proteins that are as small as 150 amino acids, but it's possible. I mean, the RNA polymerase is 5,000 amino acids in length. The RNA polymerase. The enzyme that copies DNA into RNA for the production of proteins, 5,000 amino acids in length. But just to get a short little labeling protein to form, okay? Let's look at let's look at how probabilities work first. So if you're flipping a coin and you want to get multiple heads in a row, you calculate the probability as being 1 in 2 to the power of the number you want to get in a row. Because there's a head and a tail, right? So you have 2, and if you want to get 2 heads in a row, it's 1 in 2 to the second power. Or if you want to get 3 heads in a row, it's 1 in 2 to the third power. 2 times 2 times 2. You got how that works? So, or if you want to get 8 heads in a row, it's 1 in 2 to the 8th power, which is 1 in 256. Okay? So let's look at how this works. There are 2 different kinds of bonds that can attach amino acids together, but we only want one of those 2 bonds to be present, and we want 150 amino acids. So just to get only peptide bonds in there is going to give us that number. I'm going to try to go through this real fast. Oh, what in the world? Look up. I so hate it when I can't use my remote. Give it a second. It seems to be trying to do something. Okay, the amino acids come in both It caught up with me real fast. It's like, wait, didn't get caught up with me all at one time. Well, we'll just go through it. So there are amino acids come in actually two different forms, left and right-handed forms. But in all cells, we only find left-handed forms. So there's two different forms, left and right-handed. We want 150 amino acids, but only left-handed form. We have to get that number. But, and then we want only the correct amino acids. There's 20 different amino acids. We want a correct sequence. So we want 150 of the correct type. But some amino acids can be switched out for others without affecting function. So we want to lower this number down for amino acids that are similar in chemical composition. So we can use this number instead of this big one, because some amino acids are very similar. We want to be conservative with this. But so to get a chain of amino acids that's 150 in length, we would need to multiply that number, by this number, and we get this. Just to get a one simple chain of 150 amino acids to form, it's 1 out of 10 to the 164th power. Now, there are 10 to the 80th particles in the known universe. Based on the law of probability, any odds greater than 1 out of 10 to the 50th power is impossible. And yet, to get one chain of amino acids 150 in length is 1 out of 10 to the 164th power. But the simplest cell we know of has 250 genes. The simplest cell we know of is an obligate parasite, can't live by itself, has to live inside of a host, so it has a very limited genetic component. Because of that, it has very limited genetic genome, because it uses a lot of the host proteins and things. But if it needs 250 proteins, we need to modify our probability of getting one short protein by the 250 that it really needs to perform all its life processes, and we get this number, 1 out of 10 to the 41,000th power. Darwin definitely didn't know about these probabilities when he thought life could develop. A protein could develop in some warm little pond. He didn't know, oh, what a big if. It really was. That's, oh, what a big if. That one is right here. All right, a couple other points I want to walk through with you. I'm going to have to go fast. One of them is homology. This is one of the main proofs of evolution. There's really two main proofs of evolution. One is homology. The other is the fossil record. And fossil record, best understood, is a result of the global flood. So we can just take that off the table. Fossil record is a result of global flood. Not even going to look at that. But homology refers to the similarities we see when we look at various organisms on Earth. Okay, it's what we call homology. Homology has been interpreted by evolutionists as existing due to a common ancestry. And today, again, it stands as one of the main supports of evolution, homology. For example, if you look at the various appendages of the vertebrates, amongst the mammals, 5 fingers and 5 toes is common. Amongst the birds, it's 4. You see commonalities, what we call homologies. Or at the cellular level, we see homologies. The same organelles found in various cells, right? We see a lot of cellular homology, whether it's a mitochondria, endoplasmic reticulum, same general organelles, or molecular as well. All cells use carbon as a backbone of organic compounds. They have the same macromolecules, proteins, lipids, carbohydrates, etc., and use the same system to store and use genetic information. They have the same molecules. They have the same organelles, and in many cases, they have the same big physical makeup. The term homology was coined by Sir Richard Owen. He was a British anatomist and a paleontologist, meaning fossil hunter, who is renowned for both his contributions to the study of homologies in vertebrate anatomy and for his work with fossils. He was the first person to recognize dinosaurs as distinct or different from modern reptiles. And he actually coined, in 1842, he coined the term Dinosauria. Today, however, is spoken of as almost infamous due to his strong opposition against Charles Darwin's views. Owen actually defined homology as the same organ in different animals under every variety of form and function. Owen attributed these homologies to the existence of archetypes or common design. Archetype he would define as a biological structure that are similar because they conform more or less to a pre-existing pattern, that is, to design. Owen credited God for the existence of these archetypes. In his book on the nature of limbs, Owen says, the knowledge of such a being as man must have existed before man appeared. For the divine mind which planned the archetype also foreknew all of its modifications. And we can construct archetypes with other design variations. When we think about these homologies, what they really are just similar design features. We can construct archetypes of, for example, of automobiles. What are the components that every automobile must have? That's what the archetypes really are. They're common design features. That is what we're really looking at. In 1859, Charles Darwin, however, offered a different explanation for homology. According to Darwin, similar animals possess homologies like bones because they inherited them from a common ancestor, not because they were constructed according to the same archetype. In 1892, the evolutionary biologist, again Ernst Mayr, defined homology as a feature in two or more taxa. It is homologous when it is derived from the same or corresponding feature of their common ancestor. What Darwin originally proposed as an explanation for homology became its definition. And today, homology again stands as one of the main proofs of evolution. Well, let's look at some of the problems. There are serious problems with the claim. According to neo-Darwinism, homologous structures are due to heredity, inheritance, because genes with similar sequences are unlikely to originate independently through random mutations. Gene similarity is asserted as being due to common ancestry, and they're referred to as homologous genes. In addition, homologous structures like limbs should develop through the same mechanism if they were inherited from a common ancestor. However, it has been shown that homologous structures are frequently not due to the same genes, and homologous genes are also involved with the development of vastly different structures. It's a problem. When they talk about homologies, they're seriously in conflict here. Sir Gavin de Beer was one of the foremost embryologists of the 20th century. He was a professor of embryology at University College London, a fellow of the Royal Society, and went on to become the director of the Natural History Museum in London. In 1971, he wrote a paper which he titled Homology, an Unsolved Problem, in which he states, the inheritance of homologous structures from a common ancestor cannot be ascribed to the identity of genes. What mechanism can it be that results in the production of homologous organs, the same pattern, in spite of their not being controlled by the same genes? I asked this question in 1938, and it has not been answered. Again, homologous structures, like limbs, should develop through the same mechanism if they were inherited from a common ancestor. However, structures that are claimed to be homologous frequently develop from entirely different embryonic tissues. The appendages, for example, in invertebrates, and the digestive tract, what we call the alimentary canal, also invertebrates, are derived from different tissues in the embryo. The development of the digits, fingers, and toes in vertebrates also varies considerably. Human embryos develop a thickening at the end of the limb, then programmed cell death separates the digits. By contrast, in frogs, the digits grow outwardly and independently from individual digit buds. They develop significantly different. Sir Gavin de Beer stated in his book, Embryos and Ancestors, that correspondence between homologous structures cannot be pressed back to similarity of position of the cells in the embryo, or of the parts of the egg out of which the structures are ultimately composed, or of the developmental mechanisms by which they are formed. And some final comments here from Jonathan Wells. You saw him in the clip that I showed you previously about the finches. He's a senior fellow at the Discovery Institute. He said, homology, whether defined morphologically or phylogenetically, cannot be attributed to similar developmental pathways any more than it can be attributed to similar genes. So far, the naturalistic mechanisms proposed to explain homology do not fit the evidence. And now one last thing I want to show you. Embryology, also a form of homology here, is often used to support evolution by arguing that organisms who share similarities during embryonic development are more closely related in ancestry. What was argued was that an organism basically retraces its evolutionary history through its embryonic development. It was given the phrase, the high lofty phrase, ontogeny recapitulates phylogeny. This embryonic states recap or retell the evolutionary history of the organism. That's what was asserted. And Ernst Haeckel this was Ernest Haeckel's actually most famous act of fraud. I pointed out Ernest Haeckel to you a couple times before, Ernst Haeckel. This was his most infamous act of fraud. What he had done was he had a bunch of drawings falsified. It was his view that the more similarly the organisms were in their embryonic stages, the more similar they were evolutionarily. And to prove evolution, he had a number of drawings made up that were actually fraudulent. He had, this is one of his published plates right here, showing how similar various organisms are during their embryonic stages. So humans and rabbits, chickens, tortoises, he said, look how similar they are at the exact same stage in embryonic development. These things are identical. And he had this published, had it made up and published as a way of proving evolution. But actually during his time in 1874, one of his contemporaries took him to task significantly on this issue. He sarcastically pointed out, this is Professor Wilhelm in 1874, sarcastically pointed out that Haeckel taught in Jena University, the home of the then finest optical equipment available, and so had no excuse for the inaccuracies that he published here on embryonic development. He concluded that anyone who engaged in such blatant fraud had forfeited all respect and that Haeckel had eliminated himself from the ranks of scientific research. I mean, that's pretty bad. But since then, no one really seemed to have looked at it. And these figures are still showing up in scientific books today. In fact, one of my professors at Texas Tech, when I took molecular biology, said that although there's some problems with it, ontogeny recapitulates phylogeny is still an accurate statement. He still is arguing that what Ernst Haeckel had promoted was true. Okay, but in 1997, the full extent of Haeckel's fraud was brought to light when Michael Richardson, an embryologist at St. George's Hospital Medical School of London, evaluated the embryos of various invertebrates and published the results. This, according to Nigel Hawkes. In London Times, Richardson is quoted by Nigel Hawkes as saying, "This is one of the worst cases of scientific fraud. It's shocking to find that somebody once thought was a great scientist was deliberately misleading. What he did was to take a human embryo and copy it, pretending that the salamander and the pig and all the others look the same at the same stage of development. They don't. These are fakes." What an evolutionist will do to prove his theory. As a way of closing out, I want to kind of just point something else out, okay? One of the other areas where we should be seeing homology is in chromosome number, okay? So, but animals vary widely in the numbers of chromosomes they have, but the more similar an organism was to another, the more similar their chromosome count should be. And so just as a way of closing out, I want to do this. So again, dogs have 78 chromosomes. So just so you know, you get half of your chromosomes from your mother, half of them from your father. We get 23 chromosomes from our mother, 23 from our father. Humans have a total of 46, right? So that's why all of them are even numbers, right? Dogs have 78 chromosomes. Something else with 78 chromosomes must be very similar to dog. Well, that's a chicken. So I don't know. I've not tasted dog yet. So anyone? The dog tastes like chicken? Can anyone say? Nothing? No, okay. 24 chromosomes, that's tomato. Something else that must taste very much like a tomato too then? Okay, maybe not so much. I don't know, maybe not so, I don't know. I haven't tried escargot either, so anyone? Tomato? Yeah, okay. 36, a red panda, something must taste very much like a red panda. I don't know. Oh, but that's a worm. So, okay. Chromosome number, not very good as a homology either. But let's see, 46 chromosome. What has 46 chromosomes? Okay, just making sure. I was making sure you were paying attention. Because although evolutionists may like to claim that we've evolved from ape-like ancestors, the Bible's clear on one thing, and that's who made all these things. For by him all things are created, in heaven and on earth, visible and invisible, whether thrones or dominions or rulers or authorities. All things were created through him and for him. It's to him that we owe everything. He has made all of this for us. He's watching over us. He loves us. Let me close out in a word of prayer. Father, we thank you so much for your word. We thank you for your creation, this wonderful world that you have made that speaks of your amazing creative power and your majesty. We think of the cosmos outstretched, what a glorious thing our Father is, what it is that you've made for us, the love that you've lavished upon us, making this world and all the things that we love and enjoy. Thank you, Father. We praise you. We praise the name of the Lord God Almighty. Elohim. Praise his holy name. We praise the Son, Yeshua, the Christ, the Messiah. We praise your name. We thank you for sacrificing yourself for us that my sins could be forgiven. Father, I praise you. Jesus, we thank you. Lord God Almighty. We praise the name of the Lord God. All things we ask in Christ's name. Amen.
Thank you, Chris. Give Chris a hand. We'll take a few minutes for a few questions. In the meantime, I just want to remind you again, the way we support this ministry is by a free will offering. So if you want to, would like to contribute that, the men will be up in a minute, and you can contribute either through that offering or the donation boxes in the back is available as well. So put up your hand if you have a question. I'll bring the microphone around.
Nothing hard, just easy one. Easy.
Chris, I was curious about where you were going when you're talking about wolves and dogs, and you said that wolves are purebred, and so
all right.
why all the breeds for
All
dogs?
right, she asked, though. Okay, what Gregor Mendel established there with much of his work on genetics, again, what he was doing, he would find a pure breed plant and then mate it with another pure breed plant, and when the traits of these two pure breeds were mixed together, you saw a lot of variation of these offspring. Okay, we know what a purebred organism is. That's an organism that has identical genes for the trait in question. Everybody's got to listen, because this is technical. Okay, not just the person that asks a question, whatever. Okay. A pure breed is an organism that has two identical genes for the trait in question. So a pure breed St. Bernard, every trait that is considered a St. Bernard trait, those St. Bernards have two identical genes for those traits. You get one gene from your mother, you get one gene from your father. Yeah, if you have two identical genes for the trait, you're going to give that gene to every offspring. So a pure breed is very genetically limited. They only produce pure breed. They only produce the exact same children as themselves. For every trait that they're purebred for, they can't do anything else. They have very limited genetic variability. That's what a purebred organism is. We've stripped away by back crossing an organism, by breeding it, selectively breeding it to those traits, you eventually eliminate all variability. So the process of selection is a process of reducing genetic variability. You're selecting for the specific trait that's an advantage or the one that we like, and you're reducing all the other genes through that selective process, okay? But the point I was kind of alluding to, evolutionists say, well, both creationists and evolutionists say that all the various dog breeds were isolated from following domestication of the gray wolf. So all the various dog breeds have developed from the gray wolf. Okay, evolutionists say that the gray wolf already had all of that genetic variability. It developed in the gray wolf through a long series of mutations. Okay, the creationists say God made the gray wolf with all that genetic variability, and that's where it all came from. Okay, but again, the gray wolf was a purebred. Gray wolf bred true to wolf traits. Based on our understanding of genetics and heredity, that means that every trait that bred true from the wolf, it was a genetic homozygote for. It had two identical genes for all those traits. The question is, where did the variability in these dog breeds come from if the gray wolf bred true? If the gray wolf was a purebred gray wolf, where did all that variability come from? It couldn't have come about from genetic mutations. A mutation rate that speedy would just destroy that animal in a short period of time. It's more probable that following domestication, that variability was generated because God has created all organisms with the ability to do just that. To create variability within the population to help the population adapt to the variability we see in the world. Okay? It's a big unknown. But I think it's more probable that variability we see in the dog breeds developed since the gray wolf is domesticated than that the gray wolf possessed in the beginning. It's a more reasonable explanation. Random mutations as to produce information, ludicrous. Okay? But God created organisms with an ability to perform a level of self-genetic engineering. And I could point to a number of examples. Bacteria that are parasitizing plants using weird genetic strategies and stuff. These organisms are genetically engineering themselves.
Yeah, other questions people may have? Yeah.
So to use that subject of the wolves, so Adam and Eve were purebred. Right?
They were.
And yet we have all the races, right? So you're saying the same thing, right?
Same.
Yeah.
Uh-huh.
Just two thoughts on that same line. You did not touch on. One is epigenetics, which sort of maybe touched on when you talked about I breed and show chickens, so I'm running this stuff through my head. And I know that the same species in Florida will have problems if I ship them hatching eggs from Washington and vice versa. So there's the epigenetic thing, if you could touch on that. And then also, you didn't mention anything about retrovirus. One of my species, one of my breeds is the Americana, which lays a blue egg, and it's my understanding there is an O gene, but it has to have a retrovirus insert itself into the genome at a specific place to trigger the O gene to release the enzyme or whatever it is, the blue color that's in bile that goes into the egg development. So can you touch on retrovirus and epigenetics?
No, when they talk about epigenetics, it's now well understood that not everything that makes you up the way you are is found in the DNA. That there is some information definitely outside the DNA. And it's various where that is, but so epigenetic just means genetic information, inherited information that's not found in the DNA, that exists elsewhere within the cell or in a larger scale within the organism itself. I really can't give you much on the retrovirus either, but a retrovirus, normal viruses are DNA. And it speaks to, a virus is a DNA package. Okay, so it's carrying a DNA load that's wrapped in a protein coat. It injects its DNA load into you. The goal of that seems to be to insert that into every cell in your body. So doubtful that the virus, although viruses do make us sick, they make you sick because it's trying to copy itself about 100 trillion times and insert itself into every single cell in your body. What a virus seems to be, at least the original created purpose of it, which still may be effective ongoing today, is that it's a genetic shuttle. The goal of that was to insert foreign DNA into you as probably probably help you to adapt to the local environment. But a retrovirus, instead of being a DNA package, is RNA. Normally, DNA is transcribed into RNA, then RNA is translated into proteins. And a retrovirus, the package itself is RNA, and then it undergoes reverse transcriptase, transcription back to DNA, and then inserts the DNA package into your genome. So, but okay, without some more specifics, I'm not sure I can add.
Okay, I'm going to bring the Q&A to a close here, and Chris will be available afterwards too in the back there, and you can ask him some more questions. So, Chris, again, thank you very much for the information. So, Chris presented about 6 or 7 different things that Darwin didn't know. Many more things that he could have covered. So, you ask people that question, why isn't evolution a fact? Why isn't it true? Well, what more evidence do you need than what Chris gave you here? I must also say, when I talk about topics like this, I have to admit that Darwin was an honest scientist. He wrote his book. There are 2 chapters which says if these things can be shown not to be true, my theory are wrong. Now, which scientists do you know today who would write a chapter in their book and challenge their theory? They just don't do that. He was honest in that approach. And I would say that if he were alive today, he would admit that his theory is really bankrupt. So that be as it may, let me just bring this to a close here. Next month, we're going to have another speaker, Maureen O'Kane, who we have not had here before. Her topic is rather long here. The spheres speak creation's truth. And what she's going to talk about largely is the uniqueness of our solar system and how it supports creation and not evolution. Maureen, when she was young, 25 or so, she believed in evolution, and she finally came to realize that the evolution theory did not make much sense. So she started to read the Bible, and then, the rest is history, as you'll say. And I've often said the best apologists we have are people who used to believe in evolution and now understand that it's bankrupt. It really isn't true science. And so we're going to hear from Maureen next week. There's a flyer, and again on the back, and of course this is also on the website, so you can get all the details there. And it's not very often that the speaker that we have next month is in the audience. So Maureen, I'm going to ask you just to stand up briefly and introduce yourself here. And that's, so thank you, Maureen. So she's looking forward to speaking here. She's a number of different creation groups already. So just in closing, I want to remind you that the resource tables are back there. That book that I may have mentioned a number of times, The Puzzle of Ancient Man, that's still available. It's a very, only $5, and a bunch of other books that are half priced you might look at there. Remind you that we have a lending library for those that would rather just lend the book or DVD rather than buy it outright. And we also have that Fallout DVD, which is an interview with university students by CMI people, scientists, asking them the question, why don't you believe anymore? What made you fall away from your faith? And it's a very instructive DVD. It's only 25 minutes long. If you want to listen to it, we have some on the table. Pick them up. But if you pick them up, you have to promise to listen to it, and after you've listened to it, pass it on to somebody else. Okay, so with that, remind you that the flyers for upcoming meetings are all on the back table. Brochures are back there, refreshments back there, so avail yourself of that as we break. Let me just close in a word of prayer. Father, we thank you for this time together. We thank you for the evidence that you have given us through your scripture. We know that from Romans 1, verses 18 to 20, and from Colossians 1, verse 16, that you are the creator and you have left us evidence for the truth of your word and the truth of creation, and we thank you for that. We thank you for the people like Chris and Maureen who can give evidence of that, and we just ask you just to bless them in their ministries and just bless the time around the table later. In Jesus' name we pray. Amen.